Biology, Chapter 13, Section 3, the genome. The main idea: genomes contain all of the information needed for an organism to grow and survive. Essential questions: what are the components of the human genome? How do forensic scientists use DNA fingerprinting? And how can information from the human genome be used to treat human diseases? The goal of the Human Genome Project, HGP, was to determine the sequence of the approximately 3 billion nucleotides that make up human DNA and to identify all of the approximately 20,000 to 25,000 human genes. The 46 chromosomes were fragmented, combined with vectors, cloned, and sequenced using automated sequencing machines. After sequencing, scientists observed that less than 2% of all the nucleotides code for all the proteins in the body. The genome is filled with long stretches of non-coding sequences. After sequencing the human genome, scientists began the process of identifying the genes and their functions. For organisms without large non-coding regions, researchers have identified genes by scanning the sequence for open reading frames. These contain at least 100 codons that begin with the start, end, and end stop codon. The Human Genome Project and other sequencing projects produce enormous amounts of data. Bioinformatics involves creating and maintaining databases of biological information like the one you just saw. Researchers analyze sequences looking for genes and predicting the structures of newly discovered proteins. Gene expression can be analyzed using DNA microarrays, tiny microscope slides, or silicon chips that are spotted with DNA fragments. This helps researchers determine whether the expression of certain genes is caused by genetic factors or environmental factors.
DNA microarrays give us a really useful technique for examining large amounts of genetic information in a fast and efficient manner. Take these cancer cells, for instance. First, mRNA is collected from two populations of these cells and converted to complementary DNA using a reverse transcriptase. The complementary DNA is tagged with different fluorescent molecules, differentiating the cancer cells, which are tagged red in this sample, from normal cells, which are tagged green in this sample. Then, the cDNA from the two groups is mixed. Microscope slides, or silicon chips, are dotted with DNA fragments containing all the genes, or possibly just fragments. The cells are incubated and then analyzed. Each spot on the microarray chip represents a gene. A red spot indicates that expression of a gene is higher in cancer cells compared to normal cells. A green spot indicates an expression in normal cells is higher, and yellow spots indicate equal expression in cancer cells versus normal cells. As you can see, DNA microarrays allow scientists to compare gene expression of two different samples, an important step in furthering our understanding of genetics.
So variations in the DNA sequence that occur when a single nucleotide in the genome is altered are called single nucleotide polymorphisms, or SNPs. For a variation to be considered an SNP, it must occur in at least 1% of the population. The HapMap Project. Regions of linked variations in a genome are known as haplotypes. An international group of scientists is creating a catalog of common genetic variations that occur in humans. Assembling the HapMap involves identifying groups of SNPs in a specific region of DNA. And we're going to be looking at 23andMe quite a bit because my son and I have both completed this. He has his results and I'm still waiting on mine, so that's why it will not be included in the video. But we will be taking a look at this very interesting thing. We found out my son's a carrier for cystic fibrosis, and the worst one that you could possibly have. He is a carrier. So just found a lot of interesting things out that we would have never known, and I think it very well could be a debate project. This is so cool. Has your doctor asked you or somebody in your family, or do you have a nurse in your family that works at a doctor's office, maybe mentioned to you, hey, have you had your genetic DNA tested yet? Because it really does make a difference on whether or not certain medications work for you. It's called Pharmacogenomics, the study of how genetic inheritance affects the body's response to drugs. And the benefits of this include more accurate dosing of drugs that are safer and more specific to individuals. So let me give you an idea of what they're talking about. So Prozac, everybody's heard of that. It comes in 20, 40, 60, 80 milligrams, etc. So it goes up by 20s. So I had a friend whose family member had been genetically tested for medication because they had a hard time getting anxiety and depression levels under control, and there were some other things going on, OCD, which was the case for the medicine. So this person was on like 40 milligrams to 60 milligrams of Prozac, and it seemed to work, but then not really. And everything else they had tried really didn't work. So then they came back and they said, let's do this right here and test your DNA. And maybe this isn't what works for you. And this will tell us, based on your DNA results, whether or not this drug is effective. So it came back, and actually, apparently, this was the only drug that showed to be most effective for her DNA. However, it needed high doses. So that's why it seemed to be working, but then didn't, because she was only getting around that 40 milligram, 60 milligram a day. So now this person takes, they started out at 80 milligrams, and now she is at 120, which would be excessive for a lot of people. But based on her DNA, that is what works for her. So it's kind of cool, kind of interesting. Gene therapy is a technique aimed at correcting mutated genes that cause human diseases. Scientists insert a normal gene into a chromosome to replace a dysfunctional gene.
Gene therapy has the potential to save millions of lives if we can just figure out how to make it work. Hey peeps, thanks for tuning in to D-News. I'm Trace. Gene therapy sounds like a nice, easy treatment, right? It's therapy. In some ways it is, on the macro level, but in your cells, it's a little bit invasive. In gene therapy, doctors are basically hacking the DNA of a living human. Using genetically engineered retroviruses called vectors, scientists infect human cells. The retrovirus can be programmed to carry a gene or a little bit of DNA that will overwrite the messed up mutation and make it work properly. It was first tried on a young girl in 1990, and despite some early failures, it has the potential to revolutionize treatment of genetic disorders. The journal Science describes one of the recent successes that gene therapists say was really exciting. A few children were born with metachromatic leukodystrophy, which causes a defective immune system and some brain disorders, and kids who have it usually don't live past the age of 5. Bone marrow contains stem cells. The cells normally produce red blood cells, but they can be reprogrammed using gene therapy. It's a little risky, but it can work. Taking bone marrow from these kids, doctors were able to infect the cells with a retrovirus and replace the stem cell's mutated gene with a repaired gene. Then they re-injected that back into the patient, and the fixed cells multiplied, and as of the time we filmed this, the patients are all in good condition and are heading to kindergarten at the time that others with that disease can't even speak. There may be future side effects, but they seem pretty happy with the result at the moment. I mean, I would be. Science just helped some kids! Woo! It's not just useful in children. Scientists have also used gene therapy on dogs to cure them of type 1 diabetes, with 2 of their doggy patients still alive years later. The treatment involved injecting 2 things into dogs' muscles, one gene to sense glucose and an enzyme to dictate glucose absorption. Scientists don't have to target our DNA. They can also use gene therapy to target the DNA of cancer cells. It's like they gave cancer cancer. You've seen this before if you've been following D News. A protein called CD47 is like a passport that tells your immune system not to attack a cell. Normally, cancer produces a ton of CD47. Using gene therapy on the cancer, scientists turned off that cell production and let the immune system blow it out of the sky like a decloaked Klingon bird of prey. Gene therapy is still in its infancy, but the promise of future cures for everything from cancer to genetic disorders is pretty incredible, and I, for one, cannot wait to see what this brings. What do you think of gene therapy? How do you see it being used in the future? Tell us your thoughts and keep coming back for more D News every day.
Genomics is the study of an organism's genome. This involves identifying genes and proteins produced by these genes. Proteomics is the large-scale study and cataloging of the structure and function of those proteins.